And the webinar is now live. Welcome to Ayush HardTalk. Today is Sunday, December 6th, 2020 in most parts of the world. And we are here to discuss what is scientific evidence. Today is going to be a very strong session for those of you who have been questioning this for years or months and want to know what is real scientific evidence. With us, we have three people from very different fields. The first is Professor Sastri. Prabhakar Sastri is a chemist, a metallurgist, has been consulting in metallurgy and industrial engineering, and has devised not only metallurgic processes, but the software for many factories and large-scale productions in engineering and chemistry. And he absolutely knows what statistics and evidence is. He also has a lot of personal experiences in other sciences, including what started from a food chemistry project and ended up being a cookie. And so he is a knowledgeable person in culinary arts and the science of culinary arts. So welcome, Professor Sastri. Dr. Arti Ghattaora is a clinician. She is a modern medical doctor by training and subsequently has taken Gurukul to study yoga, Siddha, meditation, Ayurveda, and has a diploma in acupuncture. She also trained in infectious disease and tropical medicine in London and has studied public health and has worked at the Wellcome Trust as a research fellow in Africa, where she worked with women and adolescents. So welcome, Dr. Ghattaora, with your knowledge of public health evidence and clinical evidence. Welcome. And we have Dr. Rammohan, who is a neuroscientist and has been doing a lot of strong work in dementia and neural development as well as neurodegeneration. He is in California and was at the Buck Institute, where he was a research associate professor, and subsequently he studied Ayurveda and became a teacher in one of the Ayurvedic colleges in California, and has been teaching not only research of Ayurveda, but obviously knows the research of the biomedical sciences. So welcome, Dr. Rao.
Thank you.
To begin, I want to ask each of you, what do you think are the challenges in scientific evidence? So to do that, of course, we need to define scientific evidence. So we'll just have you speak for a couple of minutes and we'll just keep going around because this is a true panel discussion and none of you will be presenting slides. So we'll begin with Dr. Rao. What is scientific evidence and what are some of the challenges, Dr. Rao?
So when we talk about challenges in terms of scientific evidence, the first thing we need to understand is what exactly is scientific evidence. And scientific evidence, in my opinion, is evidence or proof that serves to either support or negate a concept, a theory, an observation, or an hypothesis. So basically what we should understand is scientific evidence allows us to arrive at truth. Now, there is a misconception that scientific evidence pertains only to biomedical sciences, thanks to the COVID, but that is not so. It actually encompasses all the sciences: biological, plant, environmental, health, ecological, chemical, physical, whatever you name it. The minute you talk about arriving at truth, it means scientific evidence. So now the question is, how do we approach scientific evidence? What do we do? In my opinion, there are three approaches. Number one is an empirical approach, or what we call empiricism of scientific evidence. The second scientific evidence is intuitive approach, and I can talk about all this later. And the third is a faith-based approach. The reason why all these three constitute scientific evidence is because in all three cases, you need to gather information and make an observation. And in Vedic sciences, we call that as Shravana. Once you gather the information and observe it, you need to document that. Document the information, document the patterns, document the data. In Vedic sciences, we call this manana. And then once you have all these, now you verify it. You make it a practical observation. You practically apply that to you, whether it's to your life or whether it's to any other thing. You make a practical application through proper experimentation, nididhyasana. So whether it is empiricism or whether it's intuitive or whether it's faith-based, if you have the information observation, if you have documented that, and if you have verified that through proper experimentation, that's scientific evidence. And if none of this have been followed, or if any one of them has gone against these rules, that is where we face all the challenges.
Thank you for getting us started, Dr. Rao. I'm going to go next to Dr. Sastri and say, you know, what Dr. Rao just talked about, where we look for evidence or look for information, gather information, actually introduces what the whole issue is around evidence, which is bias, right? What kind of evidence do we see and look for? Can you talk about your ideas of what scientific evidence is and what your concerns are about its challenges, Dr. Sastri?
First and foremost, truth is not absolute. If, for example, I said that Isaac Newton's laws were absolute truth, Albert Einstein came and turned over them. So truth has a certain time. At that particular point of time, could be 100, could be 200 years, whatever it is, there is a certain truth. My problem is not with truth. My problem is with the way we arrive at data and the way we arrive at collection of the data. Most of the data, I will give you one example. Just give me 3-4 minutes. I used to do a lot of work for a company which used to be in market research. And this is a very interesting thing because we were collecting data, you know, we were sending people to the field and collecting data, whether this was in Bombay, whether it was in Calcutta, whether it was in Chennai, whether in Bangalore or whether in Delhi. For example, in one of the, I'm just giving you one example. One of them was collecting data for 400 respondents. 400 respondents over 4 towns would be 1,600 respondents. And we were collecting data for market research. Bombay produced 400 perfect data. Calcutta said, we will give you only 100, we don't want to do more than that. Chennai said, unless the questionnaire is in Tamil, we won't be able to do anything for you. Delhi data was the best because Delhi produced a total of 400, of which 100 were original, the other 300 were duplicates of the 100. Now, this is my problem. The fact that there is data, the data needs to be cleaned. Unless
How did you know? How did you know that it was duplicate data?
Because I could find that each, in fact, if I were to tell you it's 100, there were 100 clear-cut data and there were 3 of each of these with a different name and address. But the data was identical. I'm a software man, I know how to identify. Suppose there were 40 questions. All the respondents were the same. They did not even take the effort of making a change in the data. So this is my worry. And that is why I think the authenticity of the data is very essential.
Okay. Did you finish your 3 minutes? I'll come back to you, Dr. Sastri. A very important issue of falsification of data, which is, as we all know, sitting in India, and obviously in the leading journals of America too, right? New England Journal and other top journals. They can't say that we are the West and we have better data and you are not. So we know that it's an issue. Dr. Ghattaora, you've been in public health. So you and I both know in the field how much and what happens to data. So can you talk a little bit about what is scientific evidence and what are the challenges?
So I think the way that Dr. Rao has put it in terms of what scientific data really is or should be, and scientific evidence should represent, he cultivated that in a very beautiful way. From a public health clinician, I think one of the key issues that I've always had in terms of a grassroots level is when data is given to us as doctors who are trusting of the data that's given so that we can prescribe and we can actually be ready to prescribe safely. Most of the time, what we're recognizing is there are lots of publication biases that we have to overcome, and most of which we don't even know because, one, we don't have the time. So when we are given a drug, I'll take a very simple example. There was an antidepressant drug called reboxetine many years ago, and it was produced by Pfizer. And there was a lot of beautiful, there was a beautiful study done behind this where they tried it against a placebo, and it showed great effect. Again, there were 3 other, and it got published. That got published, and it was a big trial. It got published, and everybody felt very good about application. Then there were 3 other trials, which were again published against drugs which were equally efficacious in clinical usage, and it showed that it was on par. So again, it validated this idea that, okay, reboxetine is a good alternative should you have problems in prescribing this to your patients who were depressed and were not responding to other things. And you felt that there was sufficient safety in knowing this. However, it turned out that there were 7 other publications which had negative reports, which never ever got into the publication remit. So there we have the first problem with evidence, the publication bias. Now, it takes somebody like Cochrane to go and do a systematic review for that information to be gathered, to be shown that actually reboxetine is clearly not as efficacious or as safe as we deem for it to be. And the problem I felt as a clinician on the ground level is I'm prescribing things which I'm trusting for it to be safe, but however, because I don't have the time to review everything or go and evaluate how much of the negative data doesn't get into the publication remit, who do I trust? Where do I come from as a prescribing person? And hang on a minute, isn't it lives of people that we're dealing with here? Who is going to be answerable to that? Is this about an industry-driven publication bias? Is this about the fact that we have political prevalence which drives things for us to utilize it at the grassroots level? And can doctors actually have the ability to evaluate and prescribe what they feel is appropriate for a patient? So these are my questions which led me to think, as wonderful as science can be when we assess population-based data, why is it that a lot of the negative studies are withheld? I'll give you a very funny, gimmicky study which was done many years ago. Sorry. Bhaswati, you're about to say something.
Finish and then we'll come.
Yeah.
So go ahead and finish your, then we'll move.
I will just share something very anecdotal which came to my attention many years ago. And it was a study which was conducted about precognition abilities. And, you know, the scientific world, at least the scientific world that I come from, doesn't really like this idea of precognition. I mean, you know, prove it. What does it mean? Anyway, somehow a group of psychologists were able to showcase what precognition would look like. And they studied this in undergraduates. And they found that a lot of the undergraduates had these precognition abilities. Wow, brilliant, amazing. So we have undergraduates who are showcasing this brilliance. So what happened is a couple of other, so this data, by the way, got published by a well-renowned journal. It went up and there it was. Suddenly it was scientific. 3 other groups tried to replicate that same study, and guess what? The results were negative. They went back to the same journal and said, we want to actually challenge what has been shown by a previous group. Here's the data, it's negative. What happens? They said, no, we can't publish this. Simple. This is where we're at. So
thank you for sharing that.
Kind of problem with evidence from my point of view.
Okay, we're going to come back to you again. Dr. Rao, she's just talked about precognition. You're a master of neuroscience. So there's 2 issues here. One is the doctor's mental state where she wants to trust the data. And the second is the doctor learning something is true, but then later finding out it's not replicable. What do you have to say about that?
So,
Dr. Rao.
The minute you talk about issues with science, so what happens is this is where we all worry, and this is where we all go wrong. When we talk about truth, the first thing we need to understand is that any scientific evidence, okay, that verifies the truth, there is beneath that statement, there's also a statement that goes without saying, that is that it verifies the truth and it verifies the falsity of a claim. So that's very, very important that we have to understand. What it means is that whether it is a scientific evidence-based, empirical-based approach, or whether it is an intuitive-based approach, or whether it's a faith-based approach, the question is the hypothesis that you are asking, is it falsifiable? And I'll tell you why the other 2, that is the intuitive-based and the faith-based, those cannot be falsified, but the empirical-based approach can be falsified. And that's the reason why we are seeing a lot of the studies that don't get, that fail. The reason they fail is because the minute you talk about scientific evidence, you need to take into account 4 things. Number 1, is it systematized? Okay, so when you talk about systematized, the questions you're asking is there proper hypothesis? Has there been proper observation? Is there a lot of information to support that hypothesis? Is the hypothesis defined by well thought of experiments? Are there data that you have analyzed? Okay, for example now, let's say we said for this COVID, we say hydroxychloroquine treats COVID-19 infection. The question is, have you gathered all the information, literature behind this drug? Do you know its mechanism of action? Do you know its pharmaco and pharmacodynamic and pharmacokinetic properties? Okay, and then, or did you just take a simple logic analogy route? That is, since it's an immunosuppressant, and because COVID-19 features inflammation, maybe it would be a good drug for COVID-19. So that's systematized approach. Now, once you systematize the data, the question is it replicable? The minute we talk about replicability, we have to understand one thing here. Is it replicable by you? Number 1, intra, and is it replicable by others as well? Okay, so for example, and then when you talk about replicability by others, we're talking about independent verification, which is very important again. For example, you take a serum glucose analysis kit by Sigma or by Boehringer, whether it's in India or Australia or US or Africa, that serum glucose kit will work as a serum glucose kit. Now, the same thing happens whether you take a MOCA test for cognition, or whether it's a heart-lung machine, or whether it's dialysis machine. The minute you give a name to that, it works no matter where you are, in which part of the country. That is independent verification. And of course, behind that, there's also a logic. Logic, we can talk about that later. But if you have these 4 components, then it becomes very easy to verify the truth and then to see whether it can be falsifiable. And that's where I think we are having a lot, and because we have these tools now, and because we have these tools, it becomes very important to understand how we go about doing our scientific reasoning.
Okay, I'm going to come back to you with a follow-up question to this, but first I want to ask Dr. Sastri, can you talk a little bit about trusting the data? You just gave that example in Delhi where you knew software. What about other places where you might want to trust the data but can't? Can you speak to that, sir?
I thought all of you know the news today. Pfizer wants to release its vaccine in India without any trials. This is shocking because the Indian population is so different from the American population, or let us say the European population. And they've applied for permission. Shocking news. I was there see, I've had a family, my wife, my father-in-law, my mother-in-law have had cancer. I lost my wife to cancer less than a year ago. And my issue was in the year 2016, I had gone to a pharmaceutical congress because I am a man who collects data from equipment. You understand what I mean by equipment. Suppose you have a cancer patient and you have a RFID tag, and that person is going for radiation therapy. You scan the RFID tag, the patient ID is there. I can collect all the data from the equipment without anybody having to enter even one bit of the data. I talk to all the equipment that you can get in the world. So I went to this pharmaceutical congress and said, hey, here's a situation where one person like me is willing to connect to all the equipment that you have in your hospital and get data and ensure that the patient identity is safe. And then we should be able to analyze the data. So far, because all this is driven by pharmacy companies, nobody has agreed to this. And I am now talking, I lost my wife also to this. And in fact, I keep saying I didn't lose my wife to cancer. I lost her to chemotherapy. Because it is all run by pharmaceutical. Here is a person who's saying, we can connect all the equipment and give you authentic, large data. The big question there was in the conference was, what are you going to do with the data? The question is, in the conference, the theme I mentioned was, there is data. What are we looking for in this large data? I'm talking of real large data. I'm not talking of 1, 2. I'm talking in terms of millions of people. For example, the heart ECG data is available from Stanford for 1 million people, of all types of people. It is available. It is public domain. You can go and you can download it. So why is data not perceived as something which Dr. Rao will look at it in some other way, Dr. Arti will look at it in some other way, Dr. Bhattacharya will look at it some other way, I will look at it in some other point of view. Why is it that the data is not looked upon as a one point of source data rather than I collected the data? Moment you say I collected the data, I have a doubt how you collected the data. I will give you a very interesting example under 2 minutes, please. Okay. I am also a naturalist. We get data of birds in various locations. And Cornell University has got a method by which anybody in the world can post data on this. And it has been going on for the last 20 years, how the migration occurs, etc. We study this. And please let me tell you that Cornell University has identified a set of people whose data please don't use for analysis. Yes, go ahead.
Because they know that the data are not clean, right? That's what you're pointing at, Dr. Sastri. Okay. Dr. Rao, yes.
Please go ahead.
Yeah. So that's precisely the reason why I said that behind every very scientific evidence, you need to have proper systematic data. You need to verify it, and verification comes from not only your internal observation, but it has to be an independent observation. All right. And of course, you have to systematize the data. Now, let me tell you one thing. The reason why I'll agree with Dr. Sastri as well. So in my own field of neuroscience, let's take this example. I mean, 1901, okay, that is 100 years ago, the German neuropathologist, Alois Alzheimer, he made the first discovery of Alzheimer's disease. He didn't call it Alzheimer's, but then he wrote it down. 100 years later, we have numerous theories. We have already discovered the mechanisms. We have discovered the pathways. We have discovered the models, and we have created animal models of the disease. And yet, we have not come with a suitable drug till today. Just last month alone, both Roche and Eli Lilly had to pull out 2 of their drugs from phase 2 trials. So the question is now go back and look at it. There have been thousands and thousands and thousands of research papers published, peer-reviewed papers, okay? And the data is all, it's authentic data. But the question is, where did we fail? Why is it that 100 years later, we still don't have a drug for this disease, despite the fact that we have dissected the entire role of what exactly happens in the brain for a person with memory loss? So why are the drugs failing? Is the data bad? No, the data is all good there. The data has been verified. The data has been verified both by the laboratories and by independent other laboratories as well. So the question is, where else have you failed? Okay, so here is where I'm going to put a very, very provocative statement. The minute we talk about human studies, okay, what is happening is, when we create animal models, when we create cell models, we are only taking a fraction of what we call as a human. Because the minute we talk about human, we have to take into consideration not just the physical body and the symptoms, but also the mind, the mental status, the emotion, the emotional status. The question is, when you are having a cancer or when you are having a neurodegenerative diseases, you're looking at the symptoms and creating a drug. So which means you're not looking at what is happening behind, what is happening at the level of mind and emotions. And the same thing happens in the lab. When we create animal models, yes. Now remember one thing, animals by themselves don't have any neurodegenerative changes. They don't suffer from Alzheimer's, they don't suffer from Parkinson's. And yet we create animal models by forced expression of certain genes, which means that we have taken only a fraction of those things. And then we're trying to replicate that in a human model. And that's the reason why drugs have failed. Because behind every drug failure, at least in the neurodegenerative field, I'm not talking about others, but at least in the neurodegeneration sector, we know why drugs are failing, because they're only taking into account the symptoms and not the mental framework and emotional framework. So even though we have the data, it's not a complete data. And that's where we are failing.
Dr. Bhaswati, I just want 1 second.
Sure.
I want to tell you this joke today that came in the paper the other day. 2 rats were talking to each other. And one rat was asking the other, would you like to be a guinea pig for the vaccine trials? The other rat is saying, let all the trials on the humans get completed.
Yes.
Yes, I saw that one. It's a good joke. Dr. Rao, I want to ask whether or not you think that maybe we need to draw a larger framework on these studies of Alzheimer's. As you mentioned, 100 years of data, a lot of cell cultures and various animal models, because obviously we're not going to test humans and cut out their brains and see what happened with different drugs. So we have to use animals, have to. But what if we draw a larger frame of reference and use another scientific or another medical system that also sees patients and perhaps sees the mind and the brain, the manas, the buddhi, the ahankar, maybe uses a different way of looking at the brain that isn't just a summative mathematical equation of simply neurons and now glial cells, and maybe as a, according to Johnny Kepnes, there's a bunch of immunologic potentials of the glial cells, and maybe there's a couple of other meningeal functions, and that's it. What if we go beyond gene theory, cell theory, and germ theory into a different framework of reference? Let me ask Dr. Arti first, because she's nodding her head. Go ahead.
And in fact, that's what I want to also share with Dr. Rao, that I would love to hear it from him, whether he has ever evaluated why, when we talk about cultures, different geographies responding or showing Alzheimer's at different layers, how can we quantify those? What's happening in those societies, which is so different? I know we would like to probably talk about turmeric and the effects of turmeric and its evaluation and the data behind that, and how ingestion of that reduces the amount of Alzheimer's in certain subgroups in the world. But the key question, Dr. Bhaswati, which you've raised, which is why I'm nodding my head, is as a public health person who was always given this population data all the time, I always struggled with this notion that what happens to the patient experience? What about the individual experience? Because when I used to prescribe a drug to a patient for whatever disease, why was I getting different types of effects from them? What is it that was happening to them which I just didn't know about? All I was monitoring was basically, this is the evidence, that's the symptom. If we can get a handle on that symptom, we have achieved well-being at a certain level. That's all. And we never ever knew, we never went into the mind. The mind was, no, there's no mind. We just work with a brain. In the modern medical protocol, it's just the brain. The mind doesn't exist because that doesn't agree with the dogma of science. The dogma of science, from where I've been trained originally, was only talking about a very mechanistic view. Nothing to do with an evolving body or an evolving sort of structure, an organic material which has, like we're all agreeing and nodding our heads with, the mind. What happens to the mind? How do we bring this into the remit of this current dialogue of what's happening in mainstream medicine? For example, if we're talking about, sorry, Dr. Bhaswati, just 1 second. Even if we're talking about something like the vaccine, which we are going to give a vaccine to a population, do we know, do we actually understand how many people will find that efficacious, how many won't, why won't they respond? Is there an individualized perspective that we are missing on this? Yes, our science is very impressive and we love doing things which recreate our questions, but what's actually happening to the patient, the one who's receiving it? Who's looking at that? Who's looking at that?
Okay, so Dr. Arti, if I could take another medical paradigm and just place it on top of what you've just said and say that perhaps it's true that Alzheimer's is caused by excess vata over a chronic period of time in which the kapha is completely lowered and it's either created an inflammatory state because of that lowering, or that there's so much depletion in the kapha that it's creating a problem. And you understood what that meant. And then you could prognosticate by building up the kapha using healthy fats, such as ghee or such as certain grithams, and you actually found positive effect. You could never prove it because you can never do a case control study on the same person and say this person would have gotten Alzheimer's and now they didn't. But if there's a genetic effect of that person and you were to see that doing that, how would you then take those results that you see in a small number of patients and try to do a bigger study in which you explain to the scientific community and created scientific evidence on the fact that increasing kapha would actually avert Alzheimer's? Could you first talk about that, and then I'll ask the other two doctors.
So that's an interesting point, because how do we translate these sciences into a language where mainstream science can accept that this is a valid hypothesis? And it's an interesting question. I think about actually in my current study, I'm thinking about the same sort of things. How do we translate this information? Because the only thing right now when I look at individualized medicine is case study reviews. That's the only way I know to go about it, because how each individual will respond differently. And because they have a constitution, we understand that. In the traditional Indic systems, we know that each individual has a constitution, which means they will respond differently to different formulations. And it's actually the precision of the person who is actually giving the formula which will dictate whether we get the desired effect that we are talking about, which is the alleviation of that symptom. So actually, I'm not sure how we would do that yes, sorry, Dr. Rao, maybe you can take it.
Dr. Rao, please.
Let's go back one step and then we see. So Bhaswati, for your question, yes, the Ayurvedic system, very powerful. So let me go back and put all those things, the bricks that I mentioned in the beginning. Is it systematized? Yes. The Bhattacharya and the Laghutraya. I mean, you can't expect anything else. 5,000 years ago, you had these amazing books that were written. Is it replicable? Yes. It's been tried, it's been tested, it's been validated. Whether it's herbs, therapies, diet, behavior, not just 100 years or not 200 years. It's been verified for the past 5,000 years. Was it independent verification? Yes. Not just in India, but it worked. Whatever theories that they had, whatever concepts they had, whatever observations they had, whether it's Triphala, whether it is Gritas, whether it is Kashayams, it worked not just in Kerala, not just in India, it worked in Indian subcontinent, it worked everywhere else. Was there a logic behind all this concept? Yes. The logic of scientific evidence with regard to human anatomy and physiology was present in Ayurveda itself. And remember one more thing, they talked not just about the physical body, the Sthula Sarira, they talked about the subtle body, Sukshma Sharira, and they went one step further. They talked about the causal body, the Karana Sharira. So they had everything in place. And one more thing that we lacked in the sense that they went from the bedside to the bench. That's Ayurvedic science is a classic example of bedside to bench. By saying that, I'm not saying that bench to bedside is inaccurate. After all, I've been a scientist, I've been working on the bench. But what happens is bench to bedside is an inaccurate and an improper illustration of how successful translational research can be conducted. Okay, and this has been proved once by not only just by researchers like me, but even by surgeons, by so many other people, that you need to observe the patient and go back to the bench and come back to the clinic. So this fluid progression from the patient to hypothesis to experimentation and back to the patient was the true essence of translational research that existed in the Ayurvedic direction. It existed by then. And we have eons of data worth 5,000 years back to formulate all these things. And so what we did in our setting, what we did is based on the 25 plus years of work that we did at the Buck Institute where I worked before, we said, let's take Alzheimer's as an example. Alzheimer's cannot be treated with a drug. The reason being that it involves the body, the mind, and the emotions as well. So we need to take a broader approach. So even though the person working with me was a neurologist, and I tried convincing him the beauty of the science of yoga and Ayurveda, he was amenable. He was amenable to those changes. So what we did is we created a setup where patients came in and because there was no drug, but yet the patient said, what can you do for us? So what we did is based on their symptoms, okay, and based on all the comprehensive data that we collected in terms of the blood reports, in terms of their behavior, in terms of their mental faculties, we created a personalized, a comprehensive program, not a drug, but a program. Okay, the program benefited from modern science, Ayurveda, yoga, and other sciences as well. And we put all these patients on this program. And guess what? Most of the people, I would say 75 to 80% people have gone back to work. For us, that is the outcome measure. Is our program working? And the outcome measure of our program is they going back to work, they paying their bills. And it's not coming from them, it's coming from the caregivers saying that our such and such person, spouse or husband or wife or whatever, grandfather, grandmother has gone back to work or is able to pay the bills if the memory is restored. So you see what's happening? Our comprehensive program has taken into account not just the symptoms, body, but also the mind, the emotions, and everything as a whole. So it's a holistic, what we call a holistic approach, a whole body, mind, emotion approach. And that's what we need to do, not just for neurodegeneration, but for every condition, be it cancer, be it inflammation, be it COVID, whatever be it. If we take into account this entire unfractionated approach, then I think that's where we'll succeed.